now invite Dr. Randir Sood to please chair the plenary lecture by Professor Bruno Pot. Dr. Sood is the chairman, Medanta Institute of Digestive and Hepatobiliary Diseases in Medanta, the Medicity, Gurgaon. He's a member of the Indian Society of Gastroenterology and a master in endoscopy. He's widely known for his pioneering work in endoscopic intervention and is a recipient of several awards, including the Padma Shri Award for his contribution to gastroenterology in 2008 and the gold medal in 2004. His expertise lies in gastrointestinal endoscopy, gastrointestinal cancers, and hepatic diseases. Dr. Sood, may I request you to introduce our speaker, Professor Bruno Pot.
Thank you and good afternoon. I think I have the privilege of inviting a very accomplished speaker, Professor Bruno Pot, who is the research director at Institut Pasteur at Lille, France. And his research interests include probiotics and particularly lactic acid bacteria, bacterial host interactions, and health claim substantiation. And he is the appropriate speaker for this talk on probiotics and mucosal immunity interactions in early life. And he holds membership of various very important committees. And he's the president of Probiotic Research Institute, Clermont, France. He's the board member of European Scientific League of Probiotics, Belgium. And he's a taxonomic subcommittee for lactobacillus and bifidobacterium and related taxa of these. So whatever terminology you hear, he's partly responsible for it. May I have the privilege of inviting Professor Bruno Pot, please.
Thank you, Dr. Sood, for the nice introduction. So I'd like to start this presentation by thanking the organizing committee and everybody that has contributed to this fantastic trip that I had so far, and also for the very interesting program which I'm still enjoying. So today I'm not going to talk too much about my own work, but rather give an overview, and some of that part may maybe even sound a little bit philosophical. But the purpose is that we have a look at how important the interaction early in life between the microbiota and the host, so the baby, the newborn, is. And actually, there was a lot of presentations and a lot of questions that have been asked today exactly on that specific topic, and I hope to contribute with some of the answers with this presentation. So I will briefly talk about the importance of the microbiota. It has been introduced a lot already. And then just show that some of these signals which come from abroad, and that could be bacteria, but that could also be nutrition, will be important in triggering the development of the immune system. And then, of course, the question is, and that was also a question that was already raised, to what extent can probiotics actually do something? And I think it's a very important question. We don't have the answer yet, but some light is there at the end of the tunnel, of course. So let's start by saying that what we are talking about is coevolution. Coevolution between the microorganism and its host, and so immune system, which has become much more powerful and able to control that diversity in a safe way. So that it is really a cohabitation, as we call it also in France, and then it has a political meaning. It means that there is an influence from both sides. So on the one hand, the immune system is determined by, or the composition from the microbiota is actually composed or co-decided by the immune system. There is stress factors, for instance, which work through the neurosystem. A student in stress for an exam could have this diarrhea effect, which is clearly a proof that there has been, without any necessary nutritional intervention, that there is a factor of stress involved. The nutritional status has been very important, and there was a number of talks on this already today. But also all kinds of external interventions. Think about antibiotics, which clearly have an effect on the composition. On the other hand, we also know that the microbiome itself has an influence on the immune system. So it has been possible to trigger the immune system within the infancy, for instance. It is a very important factor in maturation of the immune system. The gut-brain axis has also been mentioned, so we know that what happens in the intestine would also be experienced by the brain and will also trigger some reactions there. And of course also the nutritional status will be influenced by the microbiota. And there's a lot of papers that support this. So this is a slide that you have seen in a slightly different format already, and it shows that this is not a fixed situation, but it is a dynamic situation. This correlation between the host and the microbiota, which will change over the lifetime. And what I want to focus, of course, today is on this early phase, as well as in the second phase of the pregnant woman, because it has also been said today that the pregnant woman will contribute in an active way to the development of the microbiota of the infant. So what we see is that there is, at the early stage of life, there is relatively low diversity, which might be in contrast with what we say is a healthy situation, because at the elder age, we consider bigger diversity a healthier situation. The problem is, or the fact is that in babies, newborn babies, the diversity is lower, but what is counting there at that stage is the quality of the microbiota. And this was also some of the conclusions of the last talk, where actually it is being shown that we need some bacteria, and not any bacteria which is out there, could help the baby in develop a proper immune system. But what is important is that what we see, once it has been reached age of 2, the baby has reached age 2 or 3, then the microbiota becomes relatively stable for the rest of the life, except that we see a slight increase of bacteroidetes and maybe some actinobacteria which will go down at a very late age. But what is very interesting is an observation that has been made in many papers, is that women that are carrying a baby actually see a tremendous modification of the microbiota, which is temporarily and which goes back to the normal situation after that. What we see is an increase in actinobacteria, like the bifidobacteria, but also the proteobacteria, which have been mentioned as being maybe not a positive thing to have. But it is an observation that is being done at many occasions. So how do we know about all that importance? And how about the different factors? And so the proof that we have today is probably mainly about observations when things go wrong. So it's not so much about showing this bacteria will have that effect, but what we can show is if we have a dysbiosis in one way, or we have an effect which is negative like smoking or obesity, then we see that there is an impact on the microbiota. And as a result, we can see that later on in life, there is a higher risk for the development of autoimmune diseases. There was a question about that. The development of diabetes type 2. So the idea is really that at this stage already, a lot of problems that will be present later in life is decided. And the question, of course, is what can we do to prevent the situation or to restore a situation that has been in a disbalance. So immune responses definitely are the key. And if we talk about immunity, and I'm not going to give any details about immunology in this audience, but this because I think there's many people of you who know much more about immunology than I do. I'm a microbiologist in training. But if we look at this slide, then it's obvious that this is a very complicated thing to study. There's so many factors involved. There's so many collateral factors. So if we add one bacteria, it will not influence one or two parameters that we can measure, but we really have to have a look at the complete picture. So it's complex and diverse. But still, it doesn't mean we cannot learn from what's going on. So I like to focus on some of the signals that we can see from the environment. So I already mentioned the possibility that women will change their microbiota when they are pregnant. And this is very clearly shown that this is the case in this slide. Which is from Corinne et al. in CILL in 2012, and is showing on the left-hand side of the slide the microbiota in the first trimester of the pregnancy, and then a second part of the slide shows the same women microbiota in the third trimester of pregnancy. And what you can see is that there is clear differences. So you can see that in gender hill, the more orange part, the actinobacteria, they are increased. Actinobacteria include, for instance, the bifidobacteria. And they are not linked to the intake of antibiotic, they are not linked to body mass index, and they are not linked to diabetes status of the pregnant woman, which is very important because there's all factors which have been known to be able to influence it. So this is a very stable modification. This is another representation of the same observation, where you can see the composition in trimester 3 and the composition in trimester 1. And of course, the intensity reflects the number of bacteria there with the composition. And then what I will not go through all of these microorganisms, but just focus on a few of them because they will come back in other slides. So one of them is proteobacteria, and I think we had already some discussions on proteobacteria, but importantly also lactobacillus zeae in this case. Streptococcus is also something that was mentioned, but here we're talking about salivarius, which is not necessarily a pathogen, so it's not about biogenes. So it's a normal commensal of which you also found in the mouth and which we'll found also in the intestine, but also Enterococcus faecalis. So this is the intestine. Now, we see similar things happening in the vagina, actually. So the women which are pregnant, and this is a study in which 68 women which were pregnant from the vaginal swabs have been compared to 306 samples from non-pregnant women, 60 non-pregnant women. And what is the observation, the conclusion there is that, again, there is some changes, there are some changes, there is reduced diversity, under which also an increase of streptococci and certain lactobacilli. And you can see here the non-pregnant women in the slide are in gray, and you can see here streptococcus, and if you can see it from that far, and that is lactobacillus, and you see a significant increase of these two specific organisms, type of organisms in pregnant women. You can see in this 3D plot of a principal component analysis, where you can see the pregnant women in green, and then the non-pregnant women in blue, clearly showing that there is a systematic modification of the vaginal microbiota in pregnant women, which corresponds in some extent to what we can see also on the intestinal level. And there's more. So it was already mentioned today, the importance of breast milk, the fact that breast milk is not sterile anymore. And me too, I was trained with the idea that breast milk was sterile. And actually you can see that you can easily get 10 to the 9th of bacteria per liter, not per ml, per liter of healthy, in breast milk of healthy mothers. So again, if you do the isolation of those bacteria, you find that amongst others, there is again streptococci, lactobacilli, propionibacteria, and bifidobacteria present. Now, all of these bacteria, and I will come back later on this, they all produce acid, lactic acid in many cases, or propionic acid. And I'll come back on the importance of that also in a minute. But it's more. If we look to colostrum, it had also been briefly touched, I think, today. 55.3% of the samples of colostrum that were collected in a population of women in Chile contained also lactic acid bacteria, namely Plantarum, 64%, Fermentum 16%, and Pentosus 9%. Meaning that in the colostrum, which is an actively product which is delivered by the mother for the baby to start its life in a proper way, is definitely also being provided by these beneficial bacteria. So can we talk about a program change throughout? I mean, we've seen all of the factors, the intestine of the mother, the vagina, the milk, and the colostrum. All of it has some kind of signal in one or another direction, and mostly in the same direction. So what is so particular about these bacteria? Well, they can selectively, we know about lactobacillus and streptococci, that they can selectively interact with the immune system, both the innate and the adaptive immune system. We know that they, through certain factors, they can produce lactic acid. I mentioned that already. Some of them will produce bacteriocins. Some of them will produce H2O2, which is also an antibacterial, which also has antibacterial activity. So they have this kind of anti-effective effect as well. They can assist in digestion. That's also probably for many bacteria the case, but it is definitely also the case for lactobacillus and streptococci. They are less anaerobic and more resistant to acid. This could be important because the intestine of a newborn is not so anaerobic as it is at the adult stage. So we need bacteria to colonize that are more resistant to oxygen. And of course, we also know through all the oligosaccharides which are in the milk and other bifidogenic factors which are found in breast milk, that their growth is really promoted by even the composition of breast milk. So again, I think there is a reason also why these bacteria are promoted, and that's not only physiological, but there's also immunological reasons to do that. So there is a possibility probably, because these bacteria can be produced and administered to the babies, to actually use this concept in potentially promoting the quality of the microbiota through an administration at an early life, and maybe promote a health development at that stage. So can it really work? How and when does it work? So we go back to the lab there for some answers, or at least to try to find some answers. And I found this interesting study. It was not done in our lab, but it's an interesting study where a labeled strain from Enterococcus faecalis, or Physium, was actually administered orally to mice that were pregnant. And then the mice, the baby mice, were actually taken by cesarean and were investigated for the presence of the same bacteria in the meconium. And so what was clearly obvious is that by PCR, the bacterium could be detected in all the mice that had received the bacteria and were not present in the bacteria that were not fed with the bacteria. But interestingly, the bacteria could also be cultivated, meaning that the bacteria is delivered alive. It's not just a signal that is passed on, but it's an intact and functional bacteria that is transferred, confirming what has been said earlier today, that there is really some mechanisms through probably dendritic cells or through an immune system to deliver the bacteria directly to the babies. Also, as was mentioned, there's a number of papers that show that bacteria can be isolated from the umbilical cord blood of healthy neonates and also from the placenta of the mothers, as well as in the amniotic fluid and the meconium, which I just mentioned before. So again, there is definitely an activity of bacteria to be delivered, and this efflux from the mother to the child is definitely a fact. And again, we need to understand which bacteria actually are transferred and what is the likely strategy of nature to actually promote this migration of bacteria to the baby. And if we can understand that better, if we can find some patterns in that, then I'm pretty sure we're pretty close to mimicking in children which did not have the fortunate situation of being provided these bacteria to provide them in another way. So then the question comes, of course, in order to try to answer that question, what are these signals at the surface? And again, I want to start with the co-evolutionary aspect. It's very obvious that not any bacteria will do the job. We need to go back and try to trace those bacteria which have co-evolved with their hosts. And of course, they will be species-specific. And that is something that was shown also by a very nice study that was done, and that resembles some of the studies that were mentioned earlier today, in which germ-free mice were colonized by either a typical mouse microbiota or with a human microbiota. That was the human microbiota is just being derived from a human, and some of the species within the human microbiota will, without any problem, be multiplying within the mouse intestine, and we can see quite clear differences between the mouse and the human microbiota in those mice. And so what you see here is that on the phylum level, there is not so much difference actually. So you can see here the microbiota as it has been established after inoculation from a mice. And this is the microbiota inoculated from a human. And you can see that on the phylum level, there's very little differences in composition. The ratios may be slightly different, but there's very little. If we go down in the taxonomic level, then we can see that on the genus level, especially on the genus level, there is quite substantial differences. And that is not only on the species and the genus level, but it's also reflected in the immune activation that these species can evoke. Just to clear that not all the bacteria are able to trigger a proper immune development. And so in this study, what was checked was the levels of CD4, which is tolerogenic dendritic cells, so or cells, immune cells with CD4 markers at the surface, or CD8 cells. And so what we see here clearly is that in the mice which got a human microbiome, this immune triggering is not happening, in contrast to the situation where mice did receive the normal mouse microbiota. And so the situation found in the case of human microbiota resembles very much the germ-free situation. In other words, like nothing had happened. So that shows clearly that specific microorganisms might be necessary in specific organisms to trigger that. And so what these microorganisms are was suggested also in this study, and is shown in this slide, where you can see that using the mouse-segmented filamentous bacteria, the SFB bacteria, it was actually possible to complement what was not being achieved by the human microbiome. So in an administration of the human microbiome, together, or the microbiota, together with the SFB bacteria, you had, or they observed, the authors observed, a partial, almost complete restoration of some of the immune modifications that were necessary and that were expected to happen in these early stages of life. So there is definitely a relation and a possibility between the host and the microbiota. There is definitely a specificity in that relation, and so the question is, can probiotics do the job? So to do that, I think I found this paper actually, but I intended to do exactly the same, is have a look at where we had most of the data. And the study of LGG in Finland was already mentioned earlier today. So that is a list of all the studies that mainly in allergy have been executed, in which probiotics or prebiotics were given either prenatally to the mother, either prenatally and postnatally to the mother and the baby, or postnatally intervention to the baby. So it's quite a long list, and it continues on the next page. And what is obvious there is that we need more studies with other strains. Most of the studies here have been done with LGG, which is in Finland a very common strain to be used in the field of allergy. But also you'll find some BB12, which is also a strain used a lot. So when using these strains, the results are not always that clear, because in about half of the studies, there was actually a positive effect, and about half of them, the result was not as expected. And why that is for the time being is still an open question, but it has definitely to do with the conditions probably in which the microbiota originally is, either be it at the mother or be it at the infant that is being treated. So the conclusions of that list is that in 1 out of 2 studies, there is positive parameters that can be measured, but probably we need to consider much more than only administration of probiotics and look into a number of other markers that may help to determine the efficiency or the efficacy of this administration. So this is a kind of summary of what I've been telling today, which is also a nice report published in 2013. So there is a natural way, actually, that nature is using to promote the development of a specific microbiota in the neonate. So that is through the gut microbiota. We've seen some of the things of the studies there, the results, through a change of the vaginal microbiota as well. And then, of course, during birth, there is this contamination of the fetus, which becomes a baby, and in that process will accommodate also the microbiota, which has been steered, if you want, by the mother. And that will actually give, in the optimal case, a very healthy development. If things don't go right, then the hypothesis today is that an oral application of the probiotics might actually trigger or help the colonization of the mother, and in this way also deliver some of the factors to the fetus or to the baby. But it might also not be on the bacterial level, but it might be also through signaling of the immune system. So the probiotics taken could actually change some of the parameters of the immune system, which will then be copied by the baby and which will serve after that, after birth. So when do things wrong, and can we always do this? So I like to mention this study, and it's a very, very simplified image, but I think it clears and explains very well the situation we're facing when we consider an administration of probiotics in early life. So what does it do? It does nothing to do actually with probiotics at this stage, but actually it shows the risks of administration of antibiotics. So what you can see here is in the graph is the difference between the composition of the microbiota of the infant and an adult situation. So shortly after birth, there is a huge difference, which by a normal development of the immune system and the microbiota development should actually very quickly resemble the situation of an adult, which is this situation. And that will be a guarantee for a good immunological functioning and a healthy life afterwards. However, sometimes the baby is ill and he needs antibiotics, and what you see, the antibiotics will change the composition of the microbiota, and you will see that temporarily during the antibiotic course, the difference will become bigger again. In other words, we are away from the ideal situation, but if everything goes well, and then I go back, if everything goes well, then you can see that there is a tendency to go back to the normal situation, and if everything goes well, you're still able to reach the normal situation. It could be the second antibiotic course will also have the same effect and can be, without any big problem, returned to the normal situation. But as we know that the immune system is being developed within a certain time period which is rather limited, it could very well be that the window of opportunity has been gone, and that in that particular moment there is no return to the original situation, and we will have a permanently dysbiosis, which will then lead to a non-complete immune system, or an immune system which is not completely developed, and would be the cause of many of those diseases that we see today, which are related to failing immune systems, autoimmune diseases, IBD, asthma, obesity, type 1 diabetes, atopic dermatitis, MS, all of these diseases. And that this antibiotic treatment could actually be an important factor, maybe clear from this old slide from Jean-François Bach in France in 2002 in the New England Journal of Medicine, who showed that while we were happy to see a decrease of infectious diseases since the 50s, so you can see this really for all of the infectious diseases, this is due to vaccination, this is due to antibiotic invention, this is also due to higher hygienic conditions. We see in the same period a tremendous increase of all these immune-related disorders. And of course, this is probably not by accident. And we need to ask ourselves about the possibilities that antibiotics have been, to some extent, the cause of a non-perfect immune system linked to a dysbiosis of the immune system, of the microbiota. And so this slide could easily be transformed, and this is the philosophy and the hypothesis, and that this is not real science. But some of the things that we could discuss. Is it also not so that if we look to the frequency of success of the studies with LGG, for instance, or any other probiotic in children, is it not that there might be also some windows of opportunity to administer probiotics? So in this case, could we, through the administration of probiotics, prevent that this path is taken, and can we go down this way? And so the answer to that is not there today, but it might be linked to some of the, just maybe this as a short slide, this is, again, it's the same study from Prescott, that shows you that this window of opportunity probably is situated within, let's say, the first year of life, probably within the first 6 or 7 months of life, where we can have this type of intervention. But how could it possibly work then? How could we use these windows of opportunities, and what is exactly such a window of opportunity? And so one of the papers that I recently read, and it's not a recent paper, it's from 2000, it is a recent paper, sorry, for 2014, is the epigenetic effect of human breast milk. Well, after reading through the paper, I would have said maybe the title should have been Presumed Epigenetic Effects of Human Breast Milk. But it's an interesting article to read that shows that breast milk, through a number of possible mechanisms, could actually have epigenetic effects. However, if I look to this table, and that's the list of factors that are presented in this paper, there is a number of factors like lactoferrin, prostaglandin, a number of obvious things that have been studied in the past, which now are linked to some epigenetic changes, or at least expression changes. But if you see, then a lot of them are with a B, maybe you cannot see it clearly, but a lot of them are B. And if you read the details, then they said hypothesized in humans. Again, this is a very preliminary study. It's more based on hypothesis, but I think it's worthwhile to have a look into. So these epigenetic changes, they might contribute, for instance, to the persistence of effects of microbiological regulation of the host immunity. We've seen this through a number of factors in nutrition, for instance, that these epigenetic effects are clearly there. So why cannot we hypothesize that probiotics also have this kind of possible effect? So if we accept that, then there might be some strategies if at a certain moment in time the situation has not gone like it expected, the immune system development has not finished the complete phase, then is there any possibility to reverse these kind of epigenetic changes? That is something, the next question, of course, to be answered. And this an interesting article in Nature about that as well. So I'm not going to go into details, just mention this article of the ice storm babies, which has been, which is very fascinating for me, because the authors there show, and I will, there's not enough time to go into details, but the authors show that babies that were, or mothers that were pregnant of a child during a very heavy ice storm in Canada, which raised a lot of stress, houses were destroyed. Actually, these babies had a significant different immune development and where that was linked to methylations, a completely different methylation pattern in the genome of those babies as compared to a normal group, meaning that stress conditions in the mother womb can actually change the genetics of the child, which later on can be detected at the age of 12 or 13. So my conclusions for this talk. It's clear that already in utero, the mother, the microbiota, and the baby coexist as a meta-organisms, as you can call it. And this is part of the co-evolution effect or aspect that definitely is there. The purpose is to deliver to the baby in a timely manner a commensal composition of bacteria that will make sure that the baby ages healthy. And that will have an effect on the immune as well as on the metabolic system of the baby. There is evidence, I think, that this involves epigenetic changes, although we don't know nothing about the mechanisms at this stage. And probably also that this is happening in specific time slots, which we have to try to identify and use at full. So all the mechanisms, underlying mechanisms are unknown, but I think there's a lot of potential here in India to do that kind of research, and as well as in Europe, I think we would be very happy to collaborate in identifying some of those mechanisms. So it also shows the importance of systematically in all the studies that are done with probiotics to look into the microbiome composition with very nice methods which we have available today, and use all that information also in making metadata sets in which we can draw much larger conclusions. And one of the questions that was raised today, is the microbiota in an Indian population really different from a US population or a European population? It's a very interesting question that may come from the systematic review of microbiota data. And so further insights and research may definitely be necessary to understand the epigenetic changes. So the answer finally to my question, can probiotics do the job? Yes, some will probably do it. Probably they will not be alone. They will need some assistance. And it might be that it's only possible within, in this case, for modifying the immune system at large, within a limited time window of opportunity, and which, unfortunately, we are not able yet to decide exactly, but I think we are on our way. So I'd like to end by thanking my colleagues in Lille, which have not contributed to this research. This is just literature that I picked up, but they have been supporting all of our other research activities in the past. And of course, I want to thank you for your attention.
Thank you very much, Professor Bruno. That was an excellent talk on microbiota, host interaction, and early phase of life and its effect on immune system. Now, may I invite questions for the audience, please? Yes, sir.
Dr. Pot, there has been a study in Pune in India where the mothers who were low birth weight, born low birth weight, delivered a low birth weight babies, and they then got married. Those babies grew up, were married. They also delivered preterm low birth weight babies. These babies again grew up and was married. And so this is a 3-generation epigenetic study which was done. They again delivered preterm low birth weight babies. So this is a tremendous, this was done through an MRC collaboration. The very strong data of following 3 generations and seeing that this particular effect was persisting. So at the given moment they are giving, there has been a study of the microbiota also by a Pune Institute of Basic Science, and that data is emerging, but as a large intervention, they are now intervening with the probiotics. The data is not out yet, but it's a very interesting area which you brought out, that epigenetic signatures which could be carried through generations.
Thank you for this remark. I think this is really the way to go. So I was recently attending another meeting and I heard a talk of Natasha Campbell. She's a neurologist actually, but she was intrigued by the relation between the intestine and the brain. And so when she started to study from a completely different point of view, having no microbiology background, her conclusion was that the problem started in the 50s. Probably she read the article of Bach. But when people started to use antibiotics, there was a slight modification of the microbiota, which probably was was inherited. So they passed that to their children got more antibiotics, got again some changes, that were passed to their children. And so this is the situation we face today, that probably there's in the gene somehow, some of the, if we see these huge amounts of people which are developing obesity without any very significant changes in the nutritional pattern, then there must be some causes linked to that, and that could be one of the explanations. So there's definitely the way to go to frame exactly that evolution from generation to generation.
Dr. Shinjini? Yeah.
Thank you. That was an excellent talk. I just want to understand some of the challenges that one can face while studying mechanisms. So, there's a lot of interesting data which has emerged about how the microbiome could be affecting the actual duration of pregnancy as well as low birth weight. But talking about duration of pregnancy, it's the early trimester pregnancy which is important, and that microbiome, either the vaginal or the gut, could be affecting the fetus and leading to an inflammatory syndrome and leading to early preterm delivery. So trying to understand that mechanism in a human scenario, what is it? What tissue are we studying? The placenta, the umbilical cord, the vaginal fluid, the cytokines?
Well, since we don't know what causes it, I think we have to look into everything and just try to find out what is actually active.
No, but then the placenta has a lot of noise. Placenta is, So I'm just trying to ask which would be the...
I agree, and I'm afraid I don't have the magic bullet to say this is the study you have to do. I think I would have done it before then. But the problem is the complexity, and I raised the complexity of the immune system. There's the complexity of the microbiota, as everybody who has ever looked into metagenomics data knows how much noise there is. It's very difficult to find the needle in the haystack. On the other hand, I think when we start to compare some of the results, like we have seen Streptococcus and Lactobacillus in different occasions popping up, I think we will see that when we can do sufficient data, when we collect sufficient data, do enough studies, there will be some common angles within all of those studies. And that should probably give us the clue to where we can then make some more focused research on exactly checking what is going on. So for the time being, I think we will be doomed to look into as many parameters as possible with some good hypotheses in our mind, like the ones that how can bacteria be transferred. So that's probably the mechanisms of transfer will be important. Maybe not only the bacteria, but maybe also the toxins. If we talk about a bad quality dysbiosis, then it's very probable that some toxins will be involved. Are these toxins also making it to the fetus? And in that case, how does the fetus react on it? It probably doesn't have the right metabolism to degrade the toxins yet. Maybe the liver is not completely. So this is a kind of reasonings we can use in order to set up a study and check all of these different parameters. But I don't believe in this case what you mentioned, that the microbiota is the only responsible thing. There's probably other factors that definitely involve.
It is a polygenic factor, but it's one of the factors, yeah.
What kind of epigenetic changes you see in some of these individuals, like methylation, acetylation?
Exactly, yeah. Well, we don't have a lot of tools to measure epigenetics right now, so the PacBio system sequencing is one of the few sequencing methods we have that will detect during sequencing the methylations or other modifications of the DNA. So these studies are just starting to be done. And I think once we start again to have more data, probably there will be other ways of DNA being, by proteins being covered so that they will not longer be expressed. But methylation is definitely one of the mechanisms in which research has been actively done.
Yes, please, that's the last question we can have. Yeah.
Hi, good evening, Dr. Bruno. Thanks a lot for an informative talk. I just have a very brief question about your views on the efficacy of colostrum food supplements for infants who haven't been breastfed.
I'm afraid I have no idea what the effect could be. I don't know the literature on colostrum supplemented foods. Well, maybe just out of the blue, what comes to my mind is that I personally believe that the baby and the mother have a very close link. So if we have a colostrum from a mother, it's definitely destined versus the baby of that mother, which doesn't mean that it can contain components which can be suitable for other babies. And I think nature has developed that kind of general mechanisms like oligosaccharides, which are in the breast milk, to promote a general good quality. And that will be valid. But I think there's still a specificity that cannot be ignored between a specific relation between the mother and the baby. And all of these examples that I have shown point into exactly that direction. So that will still be the optimal situation. But again, I think there might be some pluses in colostrum being considered as a very early nutrition for babies, which are in certain needs of this kind of supplementation.
Thank you very much, Professor Bruno. That was an excellent talk. Thank you very much.